As Medidata's Chief Data Officer, I've spent the last 5 years leading the technical development of much of the advanced analytics and AI that you've heard about today. I also have the privilege of giving you the last talk of the day, number 14, so thank you for being here. In this closing keynote, I'm not going to talk about specific products, but I'm going to talk about some examples of why the investments that Medidata have made into data platform, into algorithms, and to building the team of highly trained experts in AI and clinical development are good investments, not only for Medidata, but the entire industry. I'll just start with 2 quick stories, and I think you're going to be familiar with both of these stories. The first story is about a game and about a machine versus a human. So right here in Seoul in 2016, a computer algorithm named AlphaGo won 4 out of 5 games of Go against world champion Lee Se-dol. This was the first time that a computer had defeated a human expert at this level. This was a massive achievement for the AI and tech communities. Just months before, experts were saying that this type of achievement could never happen, or at earliest in 10 more years. Because Go is such a complicated game, Google experts had been trying to estimate how complex the game actually is. By one estimate, in just the 3rd move of a game of Go, there are already over 130,000 possibilities. It's truly a complex game. So the question is, if Lee Se-dol had to spend his talent and energy and dedication learning the game of Go for decades, how could a computer algorithm learn the game of Go in just a few months and defeat him? So Google's developers first trained AlphaGo with a database that contained over 30 million moves from games that human players had played at the highest level. From this data, AlphaGo was already able to achieve a level of skill that's matched by human players after many decades of playing experience. Then, the computer algorithm started playing games against itself. It played millions of games against itself and learned the patterns which would lead to success or failure. And this is essentially what AlphaGo and Google did in order to defeat Lee Se-dol in 2016. We'll come back to AlphaGo in a few moments. But the second story is related to cancer research, in particular, the breakthrough that led to the identification of triple-negative breast cancer. 20 years ago, when Medidata was first started, this form of cancer was not even known. However, at that time, many breast cancer patients were not responding to the best treatments available at the time. It was up to doctors and scientists to start noticing the commonality between many of these patients who were not responding to the best treatments of the time. And over a period of several years, it became known that 3 features were in common, which then became known as triple-negative breast cancer. Today, we know that about 15 to 20% of breast cancer patients are triple-negative with these 3 genomic features. And the question is, would today's data and algorithm capabilities been able to accelerate our identification of triple-negative breast cancer? Would we have been able to save the lives of so many and accelerate business success in the area of clinical development in breast cancer? Let me show you some evidence that suggests that it would be accelerated. So this is data that's produced from a database that's now commonly available. It's called the Cancer Genome Atlas. And based on modern machine learning and clustering analysis, we're able to see all the breast cancer patients in this database and how they cluster separately. On the right-hand side, you see a group of patients smaller than on the left-hand side of this chart. Now, when scientists went through this database and started looking at this cluster result, they knew that the algorithm was suggesting that something was different in the patients on the right versus in the group on the left. And with our knowledge today of triple-negative breast cancer, we identified that all the pink points are the patients with triple-negative breast cancer. So these are the types of insights that can be accelerated to define and identify new disease subtypes or biomarkers, and could have accelerated the path to identifying triple-negative breast cancer. Now, if you think about the world of diseases, and triple-negative breast cancer represents 15 to 20% of all breast cancers, represented by these pink dots. And you think about the universe of diseases that might not be defined by just 1, 2, or 3 features, but they could be identified by 7 features, or 100, or 1,000. The brute force mechanism of medical research from before is just not going to be sufficient to identify these patterns. So what if we take an approach like the AlphaGo team did to capture the right data in a rigorous platform, to apply the right algorithms to generate insights that will lead to scientific conclusions sooner? And maybe with the power of with computer power of today, we could accelerate these types of insights from many years down to just a few weeks or months. I'm going to share with you some examples of that acceleration that was made available through the Medidata platform. So as you've heard today, Medidata is mission-driven, and we're here to change the game of clinical research. We're investing heavily in our platform, which contains data of the highest quality. We're investing heavily in the development and adoption of cutting-edge algorithms to generate new insights from this data and make them available to you. And we've also been investing heavily building up the right teams to support you in adopting this technology and these algorithms to support you in your businesses and your science. Indeed, as you heard from Glenn this morning, we've always been a mission-driven company. We believe that making science faster, making science more accurate, is good for our clients, it's good for our patients, and it's good for our business. So we envision a future of personalized medicine that will develop eventually as more and more of these technologies are created and also used by organizations like yours. So, in the rest of today's talk, I'll touch briefly on the data platform and the precious data assets that we've compiled over our 20-year history. I'll also talk about some of the algorithms, like CSA you just heard about, that have generated insights from the available data today. And then I'll also talk a little bit more about our investment in people, the types of people that we've brought to Medidata and Acorn AI to help support you in adopting these technologies. So let's start first with data. Of course, data is the most fundamental part of any clinical trial. Every clinical trial produces data. And also, in 2019, we know that the types of data sources are more abundant. There are sensors now, there are image data. So having a data platform that is sophisticated enough to ingest this data, clean the data, integrate the data, and prepare it for generating insights is crucial. Also, in the metadata platform, we've been ingesting data from ECRFs, or from clinical trials. And we have the right to create secondary analysis based on the aggregated and de-identified data asset. So we can create signals from this data. And when you work with us, you get access to information from your own trial, and that could be operational or business information, or it could be scientific information. And you also gain access to information from the aggregated data from trials of other sponsors. This is incredibly valuable since we've already supported 17,000 trials representing the clinical trial experiences of over 5 million patients. And our data spans the globe. We have patient data from over 91 countries, and this is just our core clinical trial data asset. We touched on this earlier in an earlier keynote, but I want to show you an example of what we can do when we reuse this clinical trial data asset for a secondary purpose. So synthetic control arm is a product that we have and a service where we generate the outcomes of a control arm without recruiting a single patient. Instead, we reuse the appropriate patient data from our platform and create a synthetically derived control arm. We recently published the methodology that we use with statistical experts from the FDA, Johns Hopkins University, and several pharma. This paper is available online, or you can reach out to your Medidata or Acorn AI representative to receive this white paper. And so here you see a survival analysis, a Kaplan-Meier curve, of patients in the control arm that was synthetically derived in non-small cell lung cancer. And the question is this information correct? Is it the same as the gold standard information that would be generated in a randomized clinical trial when patients are being recruited and treated under the randomization mechanism? And so in this publication, we show that the information from the synthetic control arm matches the information from the randomized control of a previously run trial. And we did not have access to the outcome information from the randomized control before we did this exercise. So this information could be created synthetically. We could reduce the number of patients getting treated on this inferior standard of care, and we actually have a moral, and I would argue a scientific obligation to start adopting technologies like this so we get more scientific evidence with the same amount of investment in money and time. And if you consider the impact of capabilities like this, there could be a 50% reduction in the cost and time of a trial used for submission, because you'd have half the number of subjects, half the number of patients to recruit. And also consider all the use cases of a synthetic control arm. Every time you run a single arm trial, you might want to have a comparator group to make a better go, no-go investment decision at the end of the clinical trial. Or you might want to use synthetic controlled data to help design your protocol, to design your trial. Or you might want to use a synthetic control arm in lieu of other sources of external controls in the cases of accelerated approval or breakthrough therapy designation drugs. This type of information can increase the level of evidence that you bring to the table when you talk with regulators. And this is precisely why we've been collaborating a lot with the FDA. So now that we've talked about the reuse of our data, let's talk about some cutting-edge algorithms. And some of this you've seen a deep dive of, but I just want to hit some of the high points for you. Today's advanced statistical and machine learning algorithms can be used to extract new insights from your data or data on our platform. And oftentimes, these new insights could not have been identified by human experts. The last discussion from Han-Mi on CSA talks a lot about data quality. And I want to just reveal to you that our algorithm, which was developed by a team of former FDA statisticians, is not only quick, but it's also very clever. We can take as input data from any clinical trial, and every variable in that trial will be scrutinized by the algorithm. The patterns of the data will teach the algorithm what it should expect from every data point. When there is a data point that appears to break the natural pattern in your clinical trial, then we identify that data point, we flag it. And it's very surprising that when we run this algorithm on clinical trials, we find material data quality issues in every single one. Here's just one example from a locked data set. This is data where the algorithm learned a pattern, and you see each patient represented as a dot. And you see there's a pattern from the upper right to the lower left on this graph. You also will notice that there's a lot of data points that seem to be going in the exact opposite direction. What our algorithm was able to figure out was that these data all arose from one clinical trial site. This is often the case where one site will have the wires crossed on a medical device, or the training at the site was done incorrectly so that you're getting data that's exactly the opposite of what you would have expected. And indeed, upon our review of many dozens of trials across over a dozen sponsors, we have found 100% of trials have data quality issues that need to be resolved, that the human-driven best practices were not able to identify. In 100% of the trials, we found site-to-site variability in adverse event reporting that needed to be made consistent. We found in 90% of the trials that two variables in the database disagreed with each other. Think of a non-serious adverse event, which also caused death. And in 30% of the trials that we reviewed, there was evidence of site misconduct. Things like data points being made up, things like visits being fabricated, or entire patients being fabricated. Data quality is of paramount importance in clinical research. All of these data problems cause operational inefficiency and also throw off the scientific accuracy of your research. Using algorithms to find these problems before it's too late to resolve them is a key way in which you can start to improve the return on all of your clinical trials. In one particular case, Reactive Pharmaceuticals, a US-based company, they adopted the use of CSA, like Han-Mi has, and in their experience, they've been able to achieve a higher level of data quality faster and more cheaply. They also are reporting that they have happier employees because they're not doing rework. Their sites are also happier and more engaged in their trials. So they've been able to speed up the rate at which they can complete clinical trials and also increase the number of databases that they lock. In their case study, they reported that they could reduce their timelines from database lock, last patient, last visit to database lock from 30 to 60 days to just 5 days. And they've been able to double the number of database locks per year. That's millions of dollars and months of time saved for them every year. In the next algorithm, I'll talk about a key operational challenge, and that is with patient recruitment. Based on our research and some publicly available research, we estimate that about 80% of trials fail to meet their enrollment timelines. 50% of sites that are initiated in a trial only recruit 1 subject or 0. And 30% of clinical trial subjects drop out of the trial. So this is causing massive amounts of friction and inefficiency for trials. Wouldn't it be nice to utilize data that's already captured on our platform and a predictive algorithm to start scoring sites before you initiate them to help determine which sites will recruit faster or slower in your trial? That's exactly what we did in this next example. We took all of the data historically captured on our platform in a specific indication area. We linked all the historical performance of the sites with respect to recruitment and trained our algorithm. And we scored all the sites before the trial started based on the patterns found in the data. And what we saw is that the actual performance of these sites corresponded with the predictions. Here, scoring a 10 meant the algorithm believed that you would that you would recruit faster. They believed that those sites scoring 10 would recruit faster. And the sites scoring 1 would recruit slower. And the black line shows the actual performance of these sites. And indeed, the sites that scored 10 recruited at a rate about 15 times faster than the sites that scored a 1. So this is an operational example of an algorithm enabling new insights to make your site selection decisions better and resulting in faster timelines. And now I want to turn to the scientific domain. So omics data is readily available, inexpensive to collect. Many companies are collecting this. And clinical trial data is a part of your regular clinical research process. Now, if we take a new approach to combine clinical trial data with that omic data, we could think of this data set as a discovery data set. And so part of our platform integrates omics data with the clinical trial data, and then we run machine learning algorithms on top to cluster patients together based on their genomic features. And we see if any pattern of genomic features correlate with outcomes. And this is exactly how we got to the publication that Sastry talked about earlier today, where we published at the American Society of Hematology, showing that our machine learning algorithm, coupled with our data platform that links the omic data, in this case it's proteomic data, with the outcome data, identified a patient subgroup with 3 times the response rate to all other patients in the trials. This was in a rare disease called Castleman's disease, and there were 7 proteins that are correlated with the definition of the patient group that responds at 65%. Contrast that with the 3 features in triple-negative breast cancer. We're starting to enter the domain where machines can recognize the patterns much more easily than humans. And this result, which I'll show here, only took our algorithm several hours to compute Once that data was captured in our platform, it was a simple matter to get to this result. And if you review the poster, you'll see one of the figures, figure 1, is exactly a cut and paste of this output from our product. So the third part of our value proposition is our people, our team. So when we think about the AlphaGo story, it's not just a story where a computer defeated a human. The computer program, AlphaGo, was actually designed by a highly talented team at Google in the DeepMind division. They used the right data, they used the right algorithms to train AlphaGo. And so we should think of the AlphaGo victory as not as a human was defeated by a machine, but a team of humans with a machine was able to defeat a human. And so this is the kind of thinking that we are bringing to Medidata and Acorn AI to design the right data, to design the right algorithms, and work collaboratively to help our clients and to help their science learn faster, make better decisions, and ultimately serve our patients better. Between Medidata and Acorn AI, we have over 2,500 employees. And with the creation of Acorn AI earlier this year, led by Sastry Chilakuri, we've brought in many highly talented people from big pharma, from consulting, from the FDA, who are looking to guide our development forward to make the most useful products, and also to help partner with our clients so that you have the right fit-for-purpose solution for your business need. The combination of our data platform, of our cutting-edge algorithms, and our people is what make us truly unique in this industry. The platform provides the highest quality data available, clinical trial data, and you get access to not just your own, but also the data from the aggregate from other trials. The algorithms that we use are vetted, they're peer-reviewed, and they're cutting edge. And of course, the team that we employ work with the mission in mind of helping you make your clinical research faster, more effective. We've already started to receive industry recognition for this work from the FDA, from the Friends of Cancer Research in Washington, D.C. We've published at not just the American Society of Hematology, but also at ASCO repeatedly. And we're starting to win these industry awards. So I used the analogy of AlphaGo and the complexity of that game to talk about the work that we do. But of course, your work and clinical development is not a game. It is a big business. It is a serious business with patients' lives at stake. And there is a massive amount of time and money being invested in your work. Currently, based on industry statistics, there's only about a 10% success rate of clinical research programs. We are confident that as we move forward and partner together, we will be able to affect the timelines, the cost, and the success rate of our clients. So just imagine the impact on the world and your organizations if we could move from 10 years to 9. Or instead of $2.6 billion, we save 10% of that, just shaving off $260 million. Or that through the generation of better quality data or omic biomarkers, we could just double your success rate. The impact could be tremendous. Some of you may know that my wife is from Seoul. We met in New York City, but she was born and raised in Seoul. And on our visits to Seoul, I often spend a lot of time talking with my father-in-law. And recently he was reflecting on his 70 years living in Seoul and how much has changed. So he shared with me this picture of Samsung Station in 1979. And yesterday, when I got to the hotel, I took a picture of Seoul today, or yesterday. And I think this change in the skyline is dramatic enough to speak for itself. The South Korean economy, the South Korean business leaders have been very smart in leapfrogging other countries' industries to go from last place to first place over this time period. The skyline here symbolizes what's possible in the world of clinical development, where we have an opportunity now to change the way we conduct clinical development from the way we've been doing it for many decades to the way that we could do it as we look forward into the future. I believe that in South Korea, you have an opportunity to leapfrog the clinical development industry around the world, just like you've done in so many other industries. And for all of us at Medidata and Acorn AI, we'd be delighted to be your partner. 감사합니다.